session. We also have Jen Bennett-Mintz on the line. Jen is Education and Outreach Coordinator for NOAA Ocean Acidification Program, who will be helping to answer questions. For the presentation, and attendees will be in a listen-only mode. You are welcome to type questions related to technical issues or questions for the presenters into the questions box at the bottom of the control panel on the right side of your screen. We will be monitoring incoming questions and will respond to them or pose them to our speakers. We're also recording the session and will share the recordings on the CCAN website. We're excited to have Alan Barton and George Waldbusser with us today. Alan is Production Manager and Research Coordinator at Whiskey Creek Shellfish Hatchery in Elkhart, Oregon. Whiskey Creek is a major supplier of oyster larvae to shellfish farms throughout the Northwest. Alan is also Project Coordinator for the Pacific Coast Shellfish Growers Association Monitoring Program, a collaboration established between growers and members of the community. Alan also has worked over the past, worked from 2004 to 2008 as an employee of OSU's oyster breeding program, a molluscan broodstock program, as well as his crew capacity at Whiskey Creek. Alan was one of the first people to connect offshore winds, upwelling, and impacts of ocean acidification on oysters. George Waldbusser is an assistant professor of ocean ecology and biochemistry at Oregon State University. His interests broadly span marine ecology and biogeochemistry, with special interest in animal-sediment interactions, ocean acidification, and marine invertebrate ecology. He has authored 36 peer-reviewed publications. He is also currently serving on the editorial board for Journal of Shellfish Research and an associate editor of Limnology and Oceanography. Previously, he served on the Washington Governor's Blue Ribbon Panel on OA. He is also a member of the West Coast Ocean Acidification and Hypoxia Science Panel. We're going to start today with Alan, and for me as a fisherman, I think it's interesting the history of what happened with the oyster industry in the Northwest, and Alan played a key part of the history when he was working with Chris Langdon and working for Whiskey Creek. So I'm a fisherman, and I imagine as an aquaculturist, when you have problems in your lab, you're looking at bacteria or viruses or maybe nutrients or something like that. But I wonder if as we start off today, Alan, could you maybe tell us how it was that you kind of made the cognitive leap to figuring out that you weren't dealing with those sorts of problems, it was something new?
Yeah, thanks, Bruce. Yeah, we started seeing unusual mortality events probably as early as 2005 when I worked at the molluscan broodstock program in Newport. And our first thought was that these problems were caused by a marine bacterium known as Vibrio tubaceae, which was known to be pathogenic to larvae. And so we developed treatment systems targeted at removing that bacteria, and frankly felt they were pretty successful improving seed production at the hatchery. The first real major commercial problems we saw at Whiskey Creek were towards the end of 2007, when basically they produced no larvae through the late summer and into fall. I came up as a hotshot consultant, basically with the idea that I would install these treatment systems, everything would work great, and I'd just stick around for a few months until they were up and running. Obviously, it didn't work out that way. I'm still working at Whiskey Creek and still trying to understand water quality issues at the hatchery to improve production. And essentially when we turned these systems on in the beginning of 2008, they just didn't work. So we started looking at a number of the usual suspects for us. As Bruce said, we basically look at things like bacteria. We looked at our food sources, our algae, and we also looked at broodstock conditioned in different locations. a lot of the normal type stuff we would look at as biologists looking at this problem. And none of them could clearly be linked to the mortality events we were seeing in the hatchery. Fortunately for us, we had a massive mortality event in July of 2008 where we basically lost all the larvae in the hatchery within a couple of days. And I say fortunately because it actually turned out to be one of the best days ever for us. We didn't have any larvae left, so we had some time to sit around and think about different causes of mortality. I remember having Dick Feely's 2008 paper on the front table at our hatchery for several months, just getting dirty on the front table and ignoring it. But since I had some time on my hands, I sat down and read it and started thinking about carbonate chemistry as a potential cause of the problems we were seeing. And sure enough, we were right in the middle of a large upwelling event along the Oregon coast, and we saw pHs as low as about 7.6 in our hatchery. So at that point, we shifted our focus and turned away from the biological factors and started looking at carbonate chemistry. By 2009, we had initiated a monitoring program at Whiskey Creek with the help of Burke Hale's lab at OSU. We collected carbonate chemistry samples throughout the summer of 2009 and also had some real-time water quality monitoring data that we collected. We then compared it with hatchery production records, and it immediately showed us a really strong correlation between the initial water these larvae were spawned into and whether or not the larval groups ultimately survived in the hatchery. At the same time, we conducted some commercial egg development bioassays and showed that there was definitely a carryover effect from the initial egg development to the ultimate survival of larvae groups 10 to 12 days later. So that was a pretty important find from us, and at that point we really started to gain some momentum on this issue. Thanks to Burke and George, Waldbusser, and Dick Feely and others, we eventually published some of this data in Barton et al. 2012 and really started the ball rolling for the next 5 years. One of the really good things that happened for us in 2010 was that George Waldbusser came over from the West Coast, from the East Coast, and started working with us extensively at Whiskey Creek. So instead of just doing experiments in the lab, he also started doing work in the hatchery, and that was a big help in understanding what was causing the mortality in our hatchery, which George will talk about in more detail in just a minute here. The other big thing that happened in 2010 for us is we got an award from Senator Ray Cantwell's office through NOAA that allowed us to establish the Pacific Coast Shellfish Growers Association monitoring program. Essentially, this was 6 monitoring stations in areas that we cared about, 3 commercial hatcheries spread throughout the Northwest, and 3 important shellfish growing areas in Willapa Bay. This funding also allowed us to build some better instrumentation and upgrade to continuous PCO2 monitoring systems at 3 of our sites. And that really fundamentally altered the way we thought about carbonate chemistry. Instead of just looking at pH, we're now at a much more sensitive measure of carbonate chemistry to look at it in a real-time sense. If our industry has made any contribution to the field of OA research, it's that we've perhaps helped to shift the focus from thinking of OA as just a big global problem, which it is, of course, but to also look more closely at coastal regions where the impacts of ocean acidification are magnified in coastal bays and estuaries throughout the Northwest and most likely throughout the world. In July 2010, we participated in a workshop in Costa Mesa, which eventually led to the formation of the California Current Acidification Network. This was a great forum for us. It allowed guys from our industry to sit down with researchers and as well as government agencies and really put our heads together on this problem, and ultimately resulted in some benchmarks for data quality that we felt like would be biologically relevant as a group. It also helped us formalize our connection with IUS and in particular with NANUS, which really allowed us to get our data online and in front of the eyes of a lot more shellfish growers throughout the Northwest, and was a big player in helping us raise awareness of this problem, both in our industry and beyond. By summer of 2011, we had an important development at Whiskey Creek, and that Burke Hale's lab was able to upgrade our PCO2 system to a combined system capable of measuring total CO2 as well as PCO2, and we also added a Durafet pH probe, giving us a a set of several parameters from which we could calculate saturation state and display it in real time. And this is the variable most closely associated with a mortality event.
Hey, Alan, we're missing you right now, so maybe we could switch over to George for a second until you come back up, all right? I'm going to do a little housekeeping also. I'd like everybody to note that there's handouts available on the go-to control panel to download. If you're having technical difficulties, type them into the question box. But maybe we could go over to George for a second until you come back up. You could talk right over the top as soon as you're there, though, Alan. So you there, George?
Yep, there I am. Can you hear me?
Yeah, you're back, Alan.
No, that's George.
Oh, sorry.
This is Alan. I lost audio there. Can you guys hear me?
Yeah, you're back. So if you want to finish up, go ahead. We just lost you for a second.
Yeah, I'm not sure what happened there. I'm on the phone now. Sorry about that, everyone. So hopefully you guys heard me talking about upgrading systems at Whiskey Creek to a continuous TCO2 and PCO2 system. And then I was moving on to talk about Washington State's Blue Ribbon Panel on ocean acidification. That helped us gain a lot of momentum on this issue and led to the formation of the Washington Ocean Acidification Center. And importantly for our industry, it supplied funding to continue the PCSG monitoring program and also gave us some funding to do some research in the hatchery on treatment systems.
Hey, Alan, can you share your screen?
I'm sorry?
I think you need to share your screen. Can you do that still, or is your computer completely down?
I'm showing my screen being shared. Do you guys not see it?
No, we're on
It is.
George. Well, we need time.
Alan, go ahead and give it another try of sharing your screen. There we go. Awesome.
Okay. Right back now.
Yeah.
You've just moved into full slideshow mode.
Okay, so at this point I was basically just talking about the Washington Blue Ribbon Panel on ocean acidification, which formed the Washington Ocean Acidification Center and also funded the PCSG monitoring program for a few more years. And it also allowed us to work on some treatment system development in commercial hatcheries. At the same time, the Oregon legislature provided funding for continued monitoring at Whiskey Creek and supplied funding for the molluscan broodstock program to look for OA tolerance within their Pacific oyster stocks. When we built our monitoring program, I like to call it the field of dreams approach to monitoring. We basically overbuilt our system and tried to get the best data quality we possibly could, with the idea that it would attract a lot of talented researchers to our cause, and I feel we've been pretty successful in that regard. NOAA has really taken the reins of our monitoring effort, and in addition to the 6 sites I already mentioned, they've already added 3 new percolators, 2 in California and 1 in Alaska, and have also funded Burkell's lab to develop low-cost PCO2 sensors that would be deployed at 5 new sites pretty soon. So in a period of about 5 years, we've gone from basically 0 carbonate chemistry monitoring systems in important shellfish growing areas to upwards of 14 or 15. So we've gained a lot of momentum there. It's really fundamentally changed the way we think about carbonate chemistry and how it impacts our industry. And it's also brought in guys like George to help us work on this problem. So I thought I'll take a break here and give George a minute to talk to you about some of the really important research that he's done over the years that has really helped us to understand what's actually happening to larvae in our hatchery.
Can you guys hear me?
Yeah, we can.
Okay. I'm just getting set up here. It seems to be, there we go. Okay, so I think I'll talk for a little bit more than a minute, as Alan said, but first I just want to really thank a bunch of people who've really helped contribute to this work and sort of expertise at Burke Hales here at OSU. And some of the early work I did with Mark Green, I'll show you a little bit of that, which really inspired kind of the way we've been thinking about this problem as well. So it was actually, I came to OSU in 2009, and I had been working on juvenile hard clams back on the East Coast with Mark Green, and I got a phone call in early 2010 from Mark Weigert, just kind of out of the blue, and he had just spoken with Mark Green back east and said, I hear you're the guy to talk to about bivalves. So Mark kind of explained to me a little bit about what was happening in the hatchery with what they sort of were calling at the time lazy larvae when the CO2 was high, and then poor production and survival issues, and so those production failures. And so this is about the time I was just finishing up the work with Mark Green, and we were looking at juvenile heart clams just past metamorphosis. And so at this stage here, where we go from kind of a swimming larva down through this transformation to post-larval, early juvenile, and then that developmental transition out to what they ultimately start to look like a real adult clam. And so there were a couple important clues that you know were really sort of embedded in that research that I found working with Mark. One was just looking at the mortality over size. And so what I have here is first is a percent mortality percent per day for juvenile hard clams at a range of sizes, experiments that Mark had run. And so in the black squares are 0.04 aragonite saturation state, and going from gray to white up to 1.4. And so these were just looking at essentially how well they survived. What you'll notice is that there's generally higher mortality overall, even at supersaturated conditions, but that effect is really magnified at these smaller sizes, at this petavelliger stage when they first settle to the sediment. And then as they grow larger, you can see that they seem to do pretty well at these kind of really corrosive undersaturated conditions when they get up to 1 to 2 millimeters in size. So I was doing some work on calcification and growth in the same species over these same sizes. And this is basically putting together the calcification data I have to basically predict where the net zero calcification was. And so we have the shell height here, again size on the x-axis, and this is just plotted in pH here, and I have a line for about where the omega aragonite is about 1. And what you'll notice here is this line starting from about 0.5 shell height gets lower and lower. So we know that they can survive fairly low pH. These are, again, hard clams as they get bigger. And when it turns out there's this sort of inflection point where this net zero calcification crosses this omega-1, and that's about 1 millimeter. And that turns out to be a really important developmental benchmark for these juvenile clams. At that point, they actually have fully developed the mantle, and the siphons start to close, so they capture food more easily. So the important thing here was coming into this, getting contacted by the hatchery and thinking about these size-dependent, which are also temporally variable effects, and it's really related to an energetic bottleneck. Sort of importantly, as they go through this transformation from here, from the petavelliger out, essentially their feeding capture is really poor here, and they're relying on endogenous energy at this transition point here. So that kind of set the stage for thinking about the data when I started working with Burke and Alan on this. And so here's a little bit more data from some of what Alan showed you. And just to sort of hit this point home again, this is essentially showing the various metrics of growth through the larval period. And this is that omega saturation state for aragonite in the initial water. So it's the first 48 hours of spawning. And essentially what you see is if we start in the upper left, you see the initial survival. There's maybe a bit of a trend there, almost significant, but really a lot of scatter. And then you'll see early and mid-growth here in the upper right and lower left. And both of these, at least the upper right, there's really no statistical effect. We start to see some correlation between that initial water and the mid-growth, and so it's the number of days to hit these benchmarks. And then finally, where we really see the impact of that initial exposure is on that final production. So there's something really significant happening in that first 48 hours, where basically about 50, just over 50% of the hatchery production is explained by saturation state in this first 48 hours. Regardless of what happened the rest of the time for the next 2 to 3 weeks after that, this explained about 50% of that production. So we're going to get to why this would matter at this stage. Again, it's going to come back to energy and essentially calcification. So the first thing I want to point out is this is some work that actually was conducted up in the hatchery, looking at different cohorts through May and August back in 2010. This is essentially an estimate of the calcification rate, work done by my former master's student, Elizabeth Brunner. And what you'll notice here is I have a mark here for that early period, that first day or so, the calcification rate. It's in just a standardized unit of per day. And that drops quite a bit, almost an order of magnitude here over this period. These are estimated by shell weight, so they're kind of graded measures that are occurring over a couple days in time. And so if we take those values, the 0.6 and about 0.04 for this later larval stage, and we fit that to the equation for the abiotic, it's important to point out it's abiotic precipitation of calcium carbonate, we can rearrange this equation to solve for a K that's needed to support this precipitation rate in R. And so what I'm basically saying here is we don't know what the K is for these values, and we know the biology is making this happen much more quickly. So if we solve for R, what we see is that that K, that rate constant, goes up exponentially and goes up exponentially very quick as you approach 1, and that you can see that numerically in the equation. And it's clearly, we know that things calcify below 1, and so it's clearly not, it's not essentially predicting the calcification rate, but the way that I frame this and we think about it is a constraint on the system. And so really why this matters at this early stage is that about within 48 hours there's no calcium carbonate, and about 48 hours following fertilization, 80-90% of the body weight is added as calcium carbonate. I'll show you the data next on the calcification surfaces appear to be more exposed to seawater at this early stage. And until they complete this initial shell formation, they're really just relying mostly on endogenous energy. And here are just some scanning electron micrographs of Pacific oyster larvae from about 10 hours to 16 hours. You can see at 10 hours here, this is the hinge and the early periostracum here that's formed on the outside, and then basically the organism, the larvae start to sort of spackle that in, is a way to think about that, and then within about 6 hours they have a fully formed shell. So it's an incredible calcification event, and I'll show you some better data on these in a minute. So more support for this direct saturation state sensitivity. Basically, the organisms, we know this is sort of a cross-section of an adult bivalve. We know that they respire from their tissues, and their tissues encapsulate that calcification surface on the inside of the shell, and so more light carbon indicates a better control of the calcification site, a better closure from the outside environment. And we know, based on the anatomy of different taxa bivalves, that they have different abilities. So mussels, for example, can really seal that calcification surface really well. Oysters, on the other hand, that fold in the mantle where that attaches and separates the inner and outer fluid here does not seal very well. And I think anyone who's eaten mussels and oysters can sort of attest to how easily oysters will come out of a shell versus a mussel. So getting back to the isotopes here, so basically a heavier isotope signal is more reflective of seawater carbon, a lighter one is more reflective of respired carbon. So what happens is over the development of the larvae from fertilization out, what we see is a transition from heavier carbon, which this was in equilibration. If it was equilibrated with seawater, you would have about a +3 signal here for the isotopes, and that decreases over time, so indicates that anatomically things are changing. They're able to seal up that calcification surfaces better. We also see changes in the food, in the tissue here, basically. So the tissue is becoming more negative, reflecting the food. So initially, again, it's sort of indicative of that early energy, early carbon it's relying on. And I'm going to skip over this nitrogen here for sake of time. Okay. So we know the eggs really provide energy for that initial shell formation, and Burke has sort of called this the larval lunch bag. He's always great at coming up with these little analogies for all these complicated things that we tend to say as biologists. So We know how important this is, and we have some data that actually indicates some of this early egg energy lipid content becomes really important for how they grow and respond to OA. Okay, so just to bring this back to this equation again, because this is really driving a lot of this early response. So there's really two ways to alter this in the way that we see this. One is you actually pump protons or do something in this calcification surface to change the saturation state. The other is that the protein matrix that is essentially the framework for the shell, you can, in theory, change the composition of that in some way, and that's an area that we're actively working on right now to understand how that may happen in the larvae. But really the key is that the bulk of the energy goes into this organic matrix protein synthesis. If you looked at what the cost of pumping protons is, it's quite small. There's some really nice work recently done out of Donald Monahan's lab by Pan et al. And looking at sea urchin larvae and how the energy allocation under OA resulted in greater protein synthesis than basically these cross-membrane ion pumps. So one way then to think about if, to sort of get a little bit more into the science here is would slower shell formation during this period potentially mitigate or offset some of these really inherent effects we see on the Pacific oysters? And so the answer we believe is actually yes. This is some data that's in review now. Similar experiments that we've done with several different species of bivalves on the Oregon coast here. These are Olympia oyster larvae. We've actually some really great work that Matt Gray and Chris Langdon's done to culture these larvae outside of the brood chamber. We see really no physiological benefit under normal conditions of brooding. And so it turns out we can rear them outside the brood chamber, remove any of the maternal effects, and do these same types of experiments we do with the Pacific oysters at this early stage. And fundamentally, I won't get into the details of the experiments. I know several of you have seen these papers, but we're essentially separating these different effects, and ultimately what we see is there's no acute response of the Olympia oysters to this to different various carbonate system parameters. And actually what we see is you might almost see a slight decrease in normal development, which is on the left side here, under increasing aragonite, and really no effects on shell growth or calcification. So why would that be? Well, it turns out that the Pacific Olympia oysters grow and calcify much more slowly, and so this is essentially from time since fertilization. We did some really, several students in mine and Chris's lab spent a lot of time taking multiple measurements over development with the Pacific oyster here in the black and the Olympia oyster in the gray. And what you'll notice here, it's about a 5-fold difference in calcification rate. And so this happens very quickly in the Pacific oyster and much more slowly here in the Olympia oyster. A couple outliers, we haven't really quite figured out exactly what's causing those. But you'll notice it's a really much slower calcification rate. So this is how fast it's happening between these two species. And then here is actually data on the ratio of the energy triglyceride lipids versus sterile lipids. And so we're using the steriles to standardize the sort of total structural lipids, and then the energy lipids here. And again, what you see is a very rapid drop in the energy lipids in the Pacific oysters and a very, very gradual drop in the Olympia oysters. Now, it's important to note that the Olympia oyster eggs are much larger. There's much more total energy there, but there's a difference in the allocation of energy versus structural lipids. So they've clearly evolved in some way that doesn't require them to have nearly as much relative energy lipid as Pacific oyster larvae, and we think it's really related to this early shell development. Okay, so the important thing then is to think about these life history bottlenecks, and so this is where some of the challenges and then adaptation and environmental relevance starts to come in. So I really focused a lot on this first dehinge development stage. I've done a lot of work looking at clams at the second sort of transition here. Both of these are periods in time where energy is very limited. And then there's these other aspects to think about in terms of habitat and shell, and also the things that are happening during reproduction and conditioning, which is a lot of the work Chris Langdon's doing with the molluscan broodstock program. And so the important thing too is to let's not get caught in the trap of thinking exposure during these other periods don't matter. Clearly there's been some really nice work by Annelies Hediger on the Olympia oyster that shows exposure during this late veliger stage carries over into these adult stages as well. Okay, so just a few more slides here. So what's happening today is different, and put some relevance on this. This is a really nice modeling paper by Barbel-Hanisch. And essentially, the rate at which we're increasing CO2 is not allowing the Earth system to catch up to help buffer that. And so the rate at which we're doubling CO2, so this is essentially if we take the pre-industrial and double it, how quickly we do that. We're in about this yellow to green period, or yellow stage here is how fast we're increasing CO2 now in the atmosphere. And you can see that drops both the saturation state and the seawater pH. But then over time, if you stop that emission, the system comes back and stabilizes because of various buffering systems within the Earth. Now, if you increase that CO2 much more slowly, you can see we don't really change the saturation state very much, and we don't drop the pH nearly as much. Now, if we, as Alan pointed out, is if we move from this sort of global perspective to coastal zones and estuaries, we know freshwater inputs will change alkalinity, and that changes how the system is going to respond to CO2. And we also know that variables in the system don't respond linearly to increasing CO2. And so here's just a graph. If we take kind of typical summer upwelling conditions along the Oregon coast and we just alter the CO2 concentration, what you'll notice is at about 400, we're not too far off from where we start to see some chronic type effects on oyster larvae and mussel larvae. And as we increase that CO2, you can see we drop much more quickly the aragonite than we do the pH. And this delta 0.3 pH unit is a value that's been used in some meta-analysis to indicate that's about the range of pH change that's needed to see effects on bivalves. Okay, I'm going to skip through this for time, but this is some really nice work, if you haven't seen it, by Claudine Hurry, looking at frequency, magnitude, and duration, looking sort of ahead. And essentially what Claudine's shown in this paper is that increasing CO2, and this is in more sort of a little bit offshore, but increases the intensity and duration of extreme events, and the system's changing much more rapidly. And so these thresholds are all set to a saturation state of 1. If we reset that to 2, it really changes this picture, and we see that a lot of these effects are happening today, and it makes sense. 2 more slides here. One, this is some nice work that Burke Howes led, and I helped sort of work on this with him a bit. This comes from some of the monitoring data that's actually been collected at the Nakata site that Alan mentioned earlier. And what we did here with this data was essentially look at 2 environmental parameters that we know are important for oyster larvae, and this is really in particular for the Pacific larvae in Willapa Bay. And one is a thermal optimum, so we need warm conditions for those larvae to be able to reproduce and develop normally. And then we know we need sort of a threshold saturation state for them to get through that early development. And so I'm going to not show you the 2011 data. There are some gaps there. But first I want to show you the 2012. And what Burke did working with this data was to essentially subtract off of the current CO2 conditions, the current carbonate chemistry, the anthropogenic CO2 that we put in the atmosphere and has gone into the system. And so what you'll notice here is we have a thermal optimal, and the current day for 2012, the modern optimal overlap for a period of just a very short period of a few days to a week or so. If we remove that anthropogenic carbon, what you'll see is we get a really large window of time when the saturation state is favorable for Pacific oyster larvae. We go to 2013, we see a similar result. There's a smaller window, but in 2013, this window is actually quite a bit larger. So again, there's natural conditions that sort of vary normally, and essentially as we add this CO2, it changes the probability, decreases the probability of having good events and good times. It doesn't mean that they won't happen in the future, but it just decreases that probability. And finally, what's really interesting, we take 2014, the conditions were so bad naturally anyway that year that even taking off the anthropogenic carbon doesn't really change that optimal window for saturation state. And again, this is sort of as we sort of think more about these thresholds and these windows of opportunity and the variability in these coastal zones, it gets really easy to look at this and say, well, it doesn't matter, the CO2 doesn't matter, but the point is it's not a linear response. The systems have these thresholds, and they have periods and times when the system can absorb more or less CO2, and there's going to be periods when the anthropogenic carbon matters quite a bit, and potentially other years where it doesn't matter nearly as much. Okay, so essentially what I like to think about this, we go back to timing and traits in terms of thinking about how shellfish and the industry are going to adapt to ocean acidification. And so we know a lot of our lab experiments happen and look at this sort of shorter biological level of organization that occurs on much shorter time scales. If you think about it as sort of wavelengths of time, it's in the order of days or so. And as you move out, really as we start thinking about populations, communities, and evolution, it happens on these larger time scales. And so we see lots of variability that occur due to tides, diurnal net community production, storms, upwelling, and so on, that contributes quite a bit to the variability we see. And as we move in that time scale, it's a decreasing frequency and magnitude of contribution to that overall signal that we see in that location. But it also increases the permanence of that perturbation. And it's a really, I think, nuanced perspective that we need to keep moving towards to try to understand how the anthropogenic carbon will impact coastal zones. So one last slide. That's really where much of the effort has and is being put into adapting for stakeholders into these new chemistry regimes in the ocean. They're sort of dealing with these conditions on a daily basis and seeing these swings in conditions. And so some of that is anthropogenic and some of it's natural, but that's we have to really think about timing and traits for these organisms and how that fits within that variability. And then second is the hatchery is only one component of the production cycle. If we just say take oysters, for example, it's a really important one, and we know it's a really critical bottleneck. And we're doing some work in my group now looking at sort of juvenile stages and trying to give the industry growers some other tools that they can use to look at components in the field, including some of the development of the CO2 sensors we've done with my student Stephanie Smith here, working in seagrass patches in Netarts Bay, and also some work we're doing with developing techniques for the growers to actually assess fitness of the larvae and juveniles at these early stages. So I'm going to send it back to Alan now, and he's going to close it out.
Okay, thanks, George.
Okay.
Can everybody hear me there?
Yeah.
Okay. Okay, so now I'm going to circle back. As you can see, George is really doing a lot of the hardcore research that's really changing the game for us and helping explain what's actually happening to larvae as ocean acidification progresses in our region. My job is a little bit different in that regard. I always like to say that we collect data at Whiskey Creek. We don't collect data so that we can make graphs from it. We collect data so that we can make money. Our goal is, I'm not as talented a researcher as a guy like George is. My goal is to put larvae in boxes and get them out to growers throughout the Pacific Northwest. And so I thought I'd sort of talk about some of the adaptations we made as we scrambled to try to understand this problem and continue to produce larvae at our hatchery. For those of you who don't know, just for clarification, what we do at Whiskey Creek is essentially we just produce larvae. We grow them for about 3 weeks until they're 12 to 24 hours from setting, and then we ship them out to growers throughout the Northwest. They're set on oyster shell and then planted in bays, and it typically takes a couple of years for them to grow to adult size. So when we began collecting real-time data, it was directly to use that data to try to produce more larvae and supply our growers. When we started getting real-time data, we learned a lot more than we expected to. We learned there's a lot of natural variability within our bay. It's a very shallow bay with lots of eelgrass, and so we learned there's times in the afternoon with artificially high pHs due to photosynthesis in the bay. So since we had learned that egg development is very critical to the ultimate survival of larvae, we started filling our tanks in the afternoon rather than in the morning when we wanted to do a spawn. Similarly, we also learned that upwelling is driven by winds out of the north, and so when we saw strong winds begin to blow out of the north, we knew we had 24 to 48 hours before that acidified water made its way across the continental shelf and into our bay. So we would spawn like crazy. We would produce a lot of larvae, try to get them through that sensitive phase before that bad water got to Netarts Bay. And so although these are seemingly simple decisions in the hatchery, they're basically the reason that Whiskey Creek is open today. These strategies of picking our moments helped us survive 2009 and 2010 until such time as we could put in more sophisticated water treatment systems. We now have a constant pH in our hatchery year-round. These systems are actually pretty straightforward. We're just buffering with sodium carbonate. And since we've done this, everything should be fixed, right? Since ocean acidification causes pH problems and we fixed the pH, we should make lots of money. Unfortunately, the ocean's not that simple, and producing larvae in a hatchery is not that simple. And in my mind, I've really had to separate the types of mortality we see in our hatchery into at least two distinct categories. There's a direct effect of ocean acidification that affects egg development, as George has shown you. And if we buffer our water incoming to the hatchery, this problem essentially goes away. And buffering the water also does a great deal to help the growth rate of bigger larvae in our hatchery. But simple buffering alleviates this problem for the most part. However, we now understand that prolonged upwelling through the summer months leads to a lot of productivity over the continental shelf and also in Netarts Bay. And at the end of the growing season, it also leads to a lot of decomposition of organic matter, both in our bay and over the continental shelf. This leads to very low oxygen concentrations in our bay and even hypoxic events, which causes mortality in all sizes of larvae throughout our hatchery. And so we've had to work over the past few years to address these problems in our hatchery, try to improve the oxygen saturations of our incoming water, and also try to oxidize the incoming water in order to take away some of the causes of this low oxygen in our water supply. These systems have proven to be pretty effective. We've survived several periods of low oxygen out in our bay with relatively little impact on our production in the hatchery, so we feel like we've made some progress in this area, although it's certainly not 100% solved at this point. What real-time monitoring has actually done for us more than anything is helped us think beyond the end of our intake pipes and start thinking about our bay as part of a system. We know that there are local drivers of pH variability in our bay, mostly from photosynthesis of eelgrass. We also know that our bay is heavily influenced by the coastal ocean, and of course we get low pH upwelled water inputs to our bay, but we also have big dinoflagellate and diatom blooms offshore that also influence carbonate chemistry in our coastal ocean and in turn affect our hatchery. Hanging over all this entire system is the 400 part per million hairy-ass gorilla in the room, which is that there's a lot more carbon dioxide in our atmosphere than there used to be, and it's changing the way that carbon is exchanged from our surface ocean waters to the atmosphere above it. Similarly, we like to think of oxygen problems in terms of where they come from, whether they originate in the bay or out over the continental shelf. And by taking this sort of systems approach to looking at our bay, it really helps us to refine our treatment systems. We think about what the causes of these conditions are, and it helps us to come up with solutions to them in the hatchery. In my opinion, we've essentially bumped the system. We've added some carbon to our coastal ocean, and it's produced some very nonlinear responses in coastal areas throughout the Pacific Northwest and most likely throughout the world. So at this point, we should be done, right? We've addressed our pH problems in the hatchery. We've addressed oxygen problems in the hatchery. So we should be making money year-round. To some extent, we are doing a lot better. We had a successful season this year, but there are still problems coming at us. But it's only because we've picked these problems off one by one and taken a systematic approach to the problem that we can see other stressors that are still out there. For one example, I can tell you that we've had 3 major diatom blooms offshore within the last year and a half that were coincident with a downturn in production in the hatchery. Now, we don't know for sure if those blooms themselves are the cause of our production problems for brief periods of time, but we know that they're related in some way. And so by picking off these other variables, these confounding variables like pH or low oxygen, we're able to see these other stressors in isolation and then continue to work on this problem in a systematic manner. Whereas if we hadn't done that, we basically just have dead larvae and not have any understanding of what will happen. So the take-home message that I'd like to give is that, you know, the coastal ocean is significantly different than it used to be. We've fundamentally altered the way this system works. Whiskey Creek Shellfish Hatchery was started in 1978, and for about 30 30 years, they essentially warmed up the water, put food in the tank, and grew lots and lots of oyster larvae. Now that's obviously an oversimplification. This is farming. There's lots of ups and downs from month to month and year to year, but it worked for about 30 years. And as of about 10 years ago, it just stopped. And we're having to put a great deal of effort forth to produce any larvae in our hatchery. And I'm pretty proud to be a part of this industry. It's got an over 120-year history in the Northwest, and I want to keep it going for as long as we can. But it's clear to me that there's some rapid changes in our coastal ocean that are a big problem for our industry and big problem for the health of our coastal bays in general. And so I hope that we can, by getting together like this, we can start to talk about the real causes of this problem and do the really hard work of addressing it before it's too late for guys like ourselves in the oyster industry. So that's all I've got in terms of a presentation. So right now I'd like to open it up to questions for both myself and George and anyone else.
Okay, Alan. Libby Jewett has a question. She would like to know what were the conditions control set in 2015. Did Willapa Bay get any recruitment this year, or is the chances of that starting to happen again changing, or I think in general that's the question, natural conditions.
I guess I don't really know the outcome. I know there was larvae in the water this year. I don't believe there was a significant commercial set. I think there's probably people who could speak to that better than I. I do know that a lot of our customers in Willapa Bay, bought larvae from us later in the year when if they had gotten a big commercial set, they most likely would have stopped purchasing from us. The work that George mentioned, maybe you can speak to it a lot better than I can, that Burke and George have been working on together, basically talks about the window of opportunity for successful settlement of larvae in Willapa Bay and sort of shed some light on how that window is shrinking as OA progresses.
Yeah, I guess I could jump in. I don't really know what the actual data are. I know people are up there working on it. I haven't seen any of it for the sets in the last couple of years. Anecdotally, at least, from talking to Sue at the Whiskey Creek and some other folks in the industry, 10, 20 years ago, many of the growers were able to plant oyster seed late in the summer, and they simply don't seem to be able to do that anymore. So many of them have stopped actually trying to plant seed much after June, July, because they just won't survive. If they get them out early and get them through those critical stages, they seem to be okay, apparently. But after that, there seems to be some problems. I'm sure some people get it to work, but that's just what I've heard from a handful of growers who've been in the industry for quite a while. So.
We have a comment that you can call Nakota Shellfish Lab to hear about the natural sets in Willapa. I have a question. I know a lot of this work started in the Northwest, but I hear a lot of interest now from shellfish growers on the East Coast, and maybe you guys even know more about worldwide, but how are the techniques and monitoring and the things that you guys have learned, how's that moving out into the larger world of aquaculture?
You can ask George. Brad Warren and his team have been dragging us all over the place for the past several years to talk about our experiences. So we've conducted workshops in Chesapeake Bay and the Gulf of Maine. We went to New Zealand a few years ago and talked to shellfish growers. I've been to Mexico recently. Several people went over to France and talked to their oyster industry about our experiences. So we're definitely doing our best to get the word out. I think, coming from an industry perspective, it's a pretty powerful tool in that we represent jobs. And so if we get industries in different areas of the world interested in this problem, or at least interested in monitoring, it gets a lot of attention from higher ups and government that maybe wouldn't otherwise get. And so that's been really Brad's mission and his group to get the word out in that way on this problem.
Yeah, I guess I'll add. I've been to a bunch of these things with Alan, and I was in Chile a couple of years ago. I know Burke's been down there more recently. And I was part of the group in France most recently with the Ocean Conservancy and SeaWeb that put that together. There's a lot of interest, and I think the reason we're out and ahead of all this in large part is because there's been a sort of confluence of the right people with the right expertise here, and people like Burke, who's made himself very available to help people understand the chemistry and to measure the chemistry and develop instruments so that people can do that more readily. So without the monitoring, without the chemistry data, it's really hard to put the pieces together. And what we've talked a lot about in France was they've had many years of really terrible production issues out there, not quite the same as what we've seen here, but there do appear to be some disease issues, and there's a clear recognition that changing the environmental parameters in some way essentially will weaken the organism, the animals, and make them more susceptible to some diseases. And so it's not that it's a end-all to be-all, but it's certainly, I think that many in the French industry now at least recognize that there's a potential for this, for a way to create a stress on the shellfish they're growing, and might make them more susceptible to some of the other diseases that are out there.
This is Bruce again. I'd also like to say that when we first formed CCAN, there was still a little doubt in the room in the industry about what we were dealing with, and the latest surveys have shown that over 90% of the growers now recognize acidification as a problem and as the cause of what was going on, and that wasn't true when we met not that long ago. We have another question also, George, specifically with respect to infaunal species such as hard clams. Settlement to the substrate occurs at well below 1 millimeter SH. Have you examined sediment pH effects, and if so, how do they differ from a hatchery set?
Yeah, I mean, clearly the conditions at the sediment-water interface are much more corrosive and less favorable. I think the other important perspective to keep in mind is if we're thinking from an industry aquaculture perspective, these guys are looking for hopefully to get 60 to 70% survival from what they start, if that's a really good spawn. Out in the wilds, most of these invertebrates, you get less than 1% survival naturally. And because they're so fecund, that sort of survival can support some populations. You need better years sometimes to get them through. But at that sediment-water interface, conditions are clearly corrosive. We do see some work, really nice work that Mark Green led. We do see a response, and so they will settle at conditions that are corrosive. But as it gets more corrosive, fewer and fewer of those organisms will actually burrow into the sediment. So they have some ability to recognize or detect that the conditions are far less favorable. And again, a lot of these things, they can do okay as it gets more corrosive, but there's a cost associated with that. So, and again, we see the same thing with the clams. If the conditions are less corrosive or super saturated with omega or high pH, they do much better. But some always get through, and these guys have seen that at the hatchery. Even with bad conditions, you get some that get through, and we see that in our experiments too. When we get really bad conditions, there's a small handful of the numbers that'll get through. Whether that will lead to some selection down the road, it's unclear at this point, I guess. But biology is very persistent, so I think that gives me some hope.
We're going to take one last question. This one. This one comes in. It says, longer term, what are possible effects on later growth? And I know you mentioned earlier that it might result in smaller animals or potentially impact on disease resistance. But are there other things out there that you can think of, George, that the effect on larger animals?
Yeah, I mean, I think there's been a lot of work that's shown changes in growth rate for larger animals, and over time, and conditions aren't too bad. Sometimes they can acclimatize to those conditions. So, it's a stress, and it's one stress of many that's related to a lot of insults that we've created in the coastal zone. And so, this additional CO2 that we're adding to the coastal zone is only making it harder for these things to get through, in addition to any number of other impacts in various places. I mean, the thing that's really unique about netarts is it really doesn't have much of a watershed most of the year when they're growing larvae, and so we've sort of can isolate a lot of the sort of more local drivers, but many places in other estuaries, you have a whole suite of other variables that are both contributing to changes in the carbonate chemistry directly that will result in some of these stressors, or other stressors in the cells, low oxygen, pollution, and so on, or thermal sort of effects. And the challenge really is, I think, trying to understand how that variability all sums up and results in some of these longer term effects. And it's a great question, and I think it's trying to build up now from some of the mechanistic understanding we have, I think, some opportunities to do that, and also taking sort of larger scale correlative effects. But I think we have to come at the problem in both ways and try to identify it. So I don't know if that necessarily answered the question, but I think it's a really good point. There's maybe things we don't really know even yet in terms of the responses. We've done a really good job of putting things in bad chemistry water in the OA world and showing a response. And I think when we're starting to do a much better job at identifying the actual mechanisms and why things respond the way they do. So we need to do a lot more work there.
Oh, sorry. Okay, thanks to both. I was muted. Yeah, thanks to both for your presentation and perspective. If anyone has further questions about content of today's presentation, feel free to contact Alan or George directly. Their email addresses can be found in the question control panel. If you're interested in joining CCAN listserv to learn more about future updates and the acidification network, please sign up on the news page on CCAN website. You can find CCAN on Google it if you want. It's easy to find. There's an address there, a little hard to read, blue on blue, sorry. Anyhow, thanks everybody for attending today's roundtable, and we'll be having more of these coming up soon. I think the next one's going to be on sensors and the X Prize. So we look forward to that. And if you go up on CCAN, you can make sure you have all the notifications about the dates and the sign up and everything else. So it'll be sometime, whatever, October, November. We don't have the dates set yet, I don't think. Anyhow, thanks everybody for a good presentation and good questions. So good afternoon.
Thanks, Bruce. Thanks, everyone, for staying tuned.